All right. Hi, everyone. Welcome here today. So my name is Adam Jenney. I'm an infectious diseases physician and senior research fellow at the Department of Infectious Diseases at the Central Clinical School of Monash University in the Alfred Hospital. So our symposium today is entitled Unmet Needs in AMR 2023. And first, I'd like to start by acknowledging and paying our respects to the traditional custodians of the Kulin Nation and all across the world. We recognise the traditional custodians as having a unique ability to care for country and the deep spiritual connection to it. We honour elders past and present, whose knowledge and wisdom has ensured the continuation of cultural and traditional practices. We are committed to genuinely partner and meaningfully engage with Australia's traditional owners, Aboriginal and Torres Strait Islander communities. Thank you. So welcome on behalf of the Centre to Impact AMR and in particular the Clinical Unmet Needs Working Group. There are several working groups in the centre and we have met for several years now as the Clinical Unmet Needs Group, and I've been privileged to have this wonderful team of scientists, engineers and clinicians to work with. This symposium is very much the result of their ideas and effort, and I'd like to acknowledge them, as well as the centre staff and Ronya Poulis from Meeting Makers, who's helped bring this symposium to fruition. So why a clinical unmet needs or unmet needs in AMR symposium? And I think we can think about unmet needs in AMR on multiple levels. Firstly, we as clinicians focus on the clinical, where we encounter difficult AMR cases every day, with new challenges arising, such as new forms of AMR or changes in the epidemiology. On a scientific level, our understanding of AMR is constantly evolving and expanding, but these insights need to be translated to the clinic, and this is an ongoing unmet need. We also have societal unmet needs with regards to AMR. There's been a lot of work in the last decade, but we still need more recognition of the threat that AMR poses, and we need to make addressing AMR a priority on a societal level. From an economic perspective, this is always a sort of sticking point, and when we think about funding, in particular, we focus on funding for antibiotic development, and there have been some interesting steps in the last while, for example, by the UK to introduce a sort of subscription-style model for restricted antibiotics. But we need to continue to challenge how we apply different economic models, and not just to treatment, but also to other aspects of AMR, such as surveillance and prevention. Finally, there is the communication unmet need, and I think that there's very much a gap in how we communicate AMR to the broader population. So how can we better communicate? Recently, there have been initiatives by the WHO and CDC that are trying to capture patient stories, and this has been very encouraging. But we haven't seen a lot of that on an Australian level at this point. But while we've heard all of this before, I'd like to put forward that the greatest need in some ways is the need for collaboration. So we have this issue of being in our professional silos, and it's like that parable of blind people trying to approach the elephant that is AMR. The Centre to Impact AMR actually provides a perfect opportunity to move beyond some of these challenges due to the diversity of people that are affiliated with the centre and the kind of work that they're doing. Now, in addition to that, we have the Monash Partners Network, which expands the reach to a variety of clinical sites and patient populations across several major teaching hospitals in Melbourne. Given the richness of these existing networks, we wanted to actually sort of start local in our first instalment, and that's why we've invited you here today. Now, to encourage us to move beyond some of the challenges of working in isolation, we've specifically structured the symposium in sessions that incorporate an initial clinical perspective, which is really a perspective from the coalface to sort of set the scene. And then this will be followed by 2 scientific presentations regarding potential solutions to the clinical problems outlined. Finally, there'll be a panel discussion to close each session. And we've intentionally left a good amount of time to allow for questions and hopefully a dialogue, not just between the panelists, but also with you, the audience. And I think we're a small enough group for that to actually work well. So this is the overview of what we'll be doing today. And As you can see, we have 4 sessions which will run through improving diagnosis, addressing gaps in AMR treatment, optimizing our current AMR treatments, and preventing AMR. And I thank all the people that have agreed to both chair the sessions and be speakers, and thank everyone for braving the weather. As you can see, I spent hours preparing my hair this morning, and here we are with me looking like a drowned rat. So hope it can only get better from here, really. So to sort of frame each of these sessions, I'll just run through a couple of different aspects. So from a diagnostics aspect, I think despite the fact that we're sitting here in 2023 in the clinical microbiology lab, unfortunately, a lot of the time we're still relying on early 20th century technology to make our diagnoses. And here I'm talking about traditional bacterial culture techniques, for example. Not surprisingly, this leads to quite a significant delay in the time to results. And then these delays have a significant impact on care. In particular, when we think about antibiotic susceptibility, this is a big issue. So a lot of the time, we either have to end up using empiric therapy that is too broad and contributes to further AMR, or we run the risk of not providing adequate treatment for a resistant pathogen. Diagnostics can actually help address some of these concerns, and I'm sure that we'll go into a lot more detail in the diagnostics session regarding this. Now, when we talk about treatments, I think, obviously development of therapeutics is a crucial issue. And I think, we've all seen this timeline of antibiotic development, and it can kind of be described as a situation where we were victims of our own success. So you can sort of see in the mid-century there, that was the golden era of antibiotic development. They were coming thick and fast. And with this sort of success, and also the success of large-scale vaccination drives, infectious diseases were sort of deemed vanquished, and there was a lot less interest and a lot less effort put into development. As a result, by the time that we reached the 1990s, that left us fairly poorly prepared for the challenges of antimicrobial resistance that arose. And this really, is something that we encounter on a daily basis in the clinical setting, where oftentimes we have a paucity of treatments, where we're having to use old-fashioned, toxic and less effective drugs, such as aminoglycosides and even polymyxin sometimes, because there are few options. I think the corollary of all of this is that we really have to try very hard to preserve and optimize how we use our current treatments to avoid further losses. And so one of the sessions will really focus in on that. Now, fortunately, it isn't all doom and gloom. I think in the last sort of 5 to 10 years, we've been very happy to see a number of new agents reach clinical practice. The other sort of interesting thing to note about these, in particular the gram-negative agents, is that these new antibiotics are mechanism dependent. So understanding the mechanism of resistance is actually crucial to targeting the antibiotic. And I think this also speaks to this idea of synergies between diagnostics and therapeutics, where the diagnostics really do end up in a very precise way guiding the therapeutics. And this is in some ways becoming analogous to what we're seeing in the cancer space. But as someone, who's practiced infectious diseases for over a decade now, it appears that we've got this approach of, developing an antibiotic, using an antibiotic, and then breaking the antibiotic. And this is quite an endless and unrewarding cycle. And it highlights the need to move beyond this kind of cycle and into truly sort of novel therapies and branches of treatments. And here are just sort of 2 very brief examples. Obviously, phage therapy is something that has been at the forefront of our minds. In particular, as Monash is really one of the front runners in Australia in terms of phage research, and we're currently running a clinical trial at the Alfred. And here, we also see these sort of synergies between scientists and clinicians in identifying priority areas. Microbiome-based therapies have also been a significant focus, and I think there's a lot of interest in these therapies as ways of preventing or treating AMR colonization. And then finally, in the last session, we'll be talking about unmet needs in AMR prevention. And this is just a sort of slide that captures the burden of disease of healthcare-associated infections. And AMR actually makes up a huge part of these healthcare-associated infections, with our hospitals unfortunately being reservoirs of AMR. And because of this huge burden of disease and the ongoing contribution of this to propagating AMR, there are significant opportunities in improving AMR prevention. So that's the end of my opening. And yeah, thank you once again for coming in today. So what we might start moving on to now is actually the first session, which will be focusing on improving diagnosis of AMR. So thank you to Adam, Simon, and Helmut for agreeing to speak today. As you can see, we've got the clinical talk followed by 2 more scientific talks. And what I'll do is introduce the speakers as they're about to come on. We might have a couple of brief questions after each talk and then have this sort of panel discussion at the end of it. And as I said, we're sort of quite a small group, so feel free to jump in as needed. So without further ado, I wanted to introduce Dr. Adam Jenney. Adam is a clinical microbiologist and head of clinical microbiology at the Alfred Hospital. He's an infectious diseases physician within the Department of Infectious Diseases at the Central Clinical School and Monash University. And in addition to his Australian activities, Adam actually has quite a passion for global health and has strong links with the Pacific, where he's an honorary professor of medicine at Fiji National University. Over to you, Adam. Oh, so it's,
then forward.
Yeah, yeah.
Okay. Thank you very much, everybody. Thanks, Nenad, for that introduction. And I missed the rain, so that's lucky. Oh, I have to hold off for 2 minutes. Any questions so far?
Are you all able to hear him from the back?
This one. Okay. And what's that? That one. Okay. So can you hear that? Okay, great. Thanks. So I'm going to have to flick back and forth. So thank you, Nana, for the introduction. So I'm going to talk a bit about the lab, and really in front of this audience, I know many of you know about a lab, and I'm going to talk a little bit about what we do. I have no declarations of conflict of interest, but I would say that these are my own views and not represent my employers, including my employer over there, necessarily. I suppose what I'm talking about is talk about what we do in a diagnostic lab, but also I have a few requests along the way to make our lives and our approach to AMR better amongst scientists such as yourselves. So, but what I'll start off with is that we're a hospital lab, but labs form part of a tier of what we actually do. So, there are reference labs, and we probably fit somewhere between 3 and 2 there, a sort of regional specialized lab to a district hospital lab dealing with acutely unwell people. But a lot of what we want to do with diagnostics is about primary health as well. But what is the lab's purpose? First and foremost, it's to serve the patients and serve the community. It's not serving the person who orders the test. In fact, I'll get back to, it's not ordering a test, it's requesting a test. I'll get back to that as well. But so we really need to serve, that's what our aim, so is to make things better for them. And in so doing, part of what we have to do is to try and meet this burgeoning scourge of AMR, and I think we can help. We like research. We assist research wherever we can. It's very important. But a diagnostic lab, it is not our core business. It cannot be our core business. So we need to collaborate to get this done. So we want your technology. We really like it, all those lovely things that you get, and all those new drugs and all those new machines. We are absolutely keen for them. And we're not stuck at, while all that technology does come from the beginning of the 20th century and 19th century. We really are very aspirational. We like big shifts. So if you look at, in the last few years, there really has been quite a revolution in what we do. So in terms of PCR and sort of using the genome as a means of investigation and diagnosis has become right up there at the front, both in qualitative and in quantitative ways. Proteomic mass spectrometry, of course, and then I'll talk a bit about point of care testing as well. So we are very keen on that technology. I'll move forward. So But where is that technology going to go? So I'm very much going to focus about this, talk about the diagnostic lab, but I absolutely, and certainly the recent COVID pandemic has told us that diagnostics can't just stay in the lab. We've got to, and your technologies need to think about point of care testing on the home or on the street, and of course reference labs as well. We can't do everything in the labs that in our diagnostic labs that we do, but as it stands, most of, in order, you know, there's this commercial aspect to all this as well. So in order to make some, you know, it's in many of the technologies, you know, need to have some means of adding benefit to the patient and also financial benefit to the people who make them. And so they do end up focusing on getting to labs like mine. So how can we as a diagnostic lab help AMR? So look, I think the sooner I like Nenad's picture at the beginning there. You know, there is time delays, and all the time we are trying to get that diagnosis quicker and quicker, so we can tell infection prevention sooner, so they can do their work and they can get on top of AMR, because prevention, you know, it's old aphorism, prevention better than cure. Nothing it's more true than here talking about AMR. Part of our main function is that reporting of resistance itself. I mean, and that's the basis of why we're talking about this. But we have a role to do that, to give the resistances, but not all of them, so that we can hold back those broad spectrum antibiotics, so that we don't use them and don't encourage resistance in places like hospitals, which are a repository for them. And then, of course, AMI surveillance, I would argue, starts with the lab, because that's where the clinical material comes. That's where we do the diagnosis. That's where we get the organisms. And that's where we can, I mean, science is basically, you know, counting, well, looking for differences and counting. And so, and why not start with organisms and say, you know, how many do we have and what proportion of them actually are different, i.e. resistant? So I think it starts in the lab. Labs we could do a lot better. Just looking at some literature, there's you know, how in the era of AMR, you know, are labs well used? Well, probably not, actually. You know, in this study, they was saying we spend a lot of time doing unnecessary tests. I was, you know, and I can. This is absolutely true. What happens in my lab too. So people will come in with a respiratory infection. They'll say, "Why not? We'll get it all done." So you know, so half of them will have urinary cultures as well, and those with urinary tract infection, they'll get something from their sputum as well. I mean, is it so? We as clinicians and we as labs, we let this happen, and so when we could perhaps be doing something else more, you know, focused on AMR, and also, you know, a lot of our tests don't actually end up helping, you know, approach someone with an AMR problem. But even when it does, only about half the time it actually ends in a clinical interaction. So meaning that we got a result, but it wasn't acted upon. So there's a slight disconnect there too. So we need to be better at focusing what tests we do and when we do them, and also about how we get that information back and turn it into a clinical, you know, process. But there's lots of people looking at this, and this is a great paper. I enjoyed reading this about how perhaps we can be better in the labs, and you know, that and I encourage people to read this, because it looking for integrating labs more, rather than just sort of you get the results, sort of integrating labs more into the whole algorithm of diagnosis and looking at AMR. And you know, as I was mentioning before, there are all sorts of reasons why labs do not get so involved. There's lack of awareness. There's actually, you know, fear of starting new tests. There's a whole lot of barriers to starting new tests, which I'll talk about in a minute. And they, you know, it needs advocacy. So we need probably clinical groups. So IDSA here advocating for the use of getting labs to do and getting people who would request the test to get them to request the test in order to best help their patients. So what do we actually do? I'm going to skip through this very quickly, but if you're ever answering the College of Pathologists exam, you answer this question by saying, well, we have the pre-analytical and with the analytical and with the post-analytical. But I mean, what it is, the only purpose I want to say of this is that it is really very old-fashioned what we do. And in fact, what we have processes to get a sample safely to the lab, safely for the people who are carrying it, but also safely for that we get a proper result, so it's not contaminated. And then we register it, and then we have to divide it, and then we have to send it to a bench over there, and then we have to set it up, so-called. Then we have to stain it, and then we have to culture it, and then we have to wait, and then we have to read it, and then we have to reculture it, and then we have to wait. And then we have to do the susceptibilities, which we call senses, and then we might do PCR, and then we might freeze it, and then we might unfreeze it and send it off to a reference lab. So, I mean, there's all sorts of processes in there, and I don't have answer to make that better. And I look at my lab, and there's this glass window, and everything comes in through that glass window, and it has not changed for 60 years. I don't have an answer. But also then we have to send it off. So the purpose is to get that result to the ordering clinician in order for them to do something good with their patient. And also in terms of talking about surveillance, we might have to send it to a higher power like the department or one of their representatives. So we are, do we always get the right answer? Well, we're very keen to get the right answer. That's our purpose to being there, but we try very hard to get the right answer, but we really like to have more and more new technology to try and be better at getting that right answer. And so QC and QA, it's our friend. We spend a lot of time doing it, and that's in order so that we have high quality results. We can only do what we're licensed to do. We can only, and we need to get credentialing both in terms of staff and in terms of machinery to do that. And we need to keep working at keeping those licensing. This takes up a lot of time. And so what I'm, however, I'm a big fan of it because I think every year we get accredited, we find something and we do it a bit better the next year, and I like that. But in terms of finding new things, that'd be really good for this, this audience to know what we do, so that the tests that come along, the technology needs to be in a form that we can use, and we're very happy to take it on, but it needs to fit all this regulation. There's a lot of regulation to keep it safe and to keep the results true. So there's lots and lots of unmet needs, and I was thinking about what have we done that may have started along this way. So infection prevention, our tests are getting more sensitive and hopefully a bit more specific, but and I talked about turnaround times, getting that, that outbreak result out quicker, recognize, I think that's important. But remember, PCR has taken over a whole lot, particularly of the virology. If you went to Vidril, so 30 years ago, so the Victorian ID reference lab, 35 years ago, there were benches and rooms full of cell culture baths, which people would come along and try not to get contaminated and put virus in and watch for cytopathic effect. They are all gone and replaced by a PCR machine. In terms of bacterial diagnosis of resistance, we're able to do it much quicker from the plate to get that resistance known. But there's the tension between the phenotype and the genotype. Is the gene being expressed? It's there, but is it being expressed? The phenotype, well, it looks like it's being expressed on the plate, but does that correlate to the clinic? None of this is perfect. So in terms of antimicrobial stewardship, we can't provide every drug-bug interaction. And we have UCAST and CLSI, they give us the guidelines, they give us the rules, they give us the best way of doing this, within the limitations of numbers and obviously the most important pathogens are there, and we can reflect on it. And our automated speedy platforms are great, but they're limited. And the old-fashioned broth dilution, you can do it for anything, but it takes lots of energy and human resources and time. But there's a shift back to it when you really want to get the answer of that critical result. So that's a very old technology which may be coming back as well. And surveillance. So we could do much more. And every day we throw away countless organisms into, you know, which could be of some use. It's because of space, because of freezer space, because of time to freeze them. But I think you know, these collaborations are very important to get that surveillance answer. I would add to that when we're looking at genomes, you know, you're looking at these massive whacks of data that you have, I would say that we have IT limitations before, you know, in getting those enormous amounts of, you know, sequence. I mean, it's convenient to send. You don't have to put it in a taxi, but you can send it. But remember, so does anyone know who Joseph Bazalgette is? Does anyone know? It's a question. There's a prize. Because no one's put up the hand. I don't have a prize, but I would go. So this fellow was, after the Great Stink of London of 1858, he was commissioned to rebuild the sewers of London. And he came up with a very clever design to make them flow, to avoid the river, to fix it. But he did two things, which was two things. He looked at London, he said there was a population of about just over 2 million. And he said, this isn't going to stay like this. So he did two things. So he doubled the size of the pipes, and then he doubled it again. And so in fact, they're still, much of them still functioning, even though they built up, and the population is now 11 or 12 million. So they're only now just failing. So I say with IT limitations, whenever you put in your budget for IT stuff, double it and then double it again. So what do we do? So we want to reverse this. Because it's an important question. What's best for the patients and community. I'll skip through that because what I want to do is some final thoughts. So one thing I said I alluded to before is pathology stewardship. So we could spend a lot of time doing more useful things than doing what we do. We do a lot of repeated tests. And this is across all pathology, and it's not the worst in microbiology, because we regard it as a request. It's not an order. It's not for a martini. I'm not going to bring that. I can actually cross it out and say. And so we actually spend quite a bit of time stopping tests. But other labs which churning through with automation, they do a lot of unnecessary tests, which we could spend time doing other things. Just in terms of equity, I mean, we are very lucky in what we have available to us, but 80% of the labs, don't have anywhere near this. And I think when this group here should look to that as well with their solutions, I think that's very important. Talking about data transfer, I mentioned that. We've also got to think about when the electricity goes out. There's a whole bunch of people who have power insecurity around the world. And so in those technologies, the stuff that when you just need a bit of sunlight and a dropper, they're very good. And I think they should be pursued. And, everything is finite. Remember that when you put in something new, it's going to push something else out. And we can't just add to and add to and add to. And the clinician colleagues don't understand that, but they you do lose something when you do it. So I will finish there, and I'm sorry I've gone a little bit over time. So thank you very much.
Yep. Excellent. Thanks, Adam. That was a wonderful introduction in setting the scene. Do we have any questions? No? Okay.
Adam, the approval and regulatory pathway for diagnostics is very different to a new antibiotic, for example, new medicine. And I often find that it's an easier, shorter pathway, and they may not have done the kind of clinical impact studies. And, that to me seems like a real limitation of these new fancy whiz-bang tests coming through, but we actually don't have an understanding of what their clinical impact is. Do you have a thought on all that?
Yeah, absolutely. So and a technology will come along and suddenly there's a whole bunch of Me Too's as well. And it's the low barrier to get over. And we are bombarded with use this and use that, but we need, what is our clinical need of this? And we need to find that. And that's where we need clinicians to tell us what to do. But we also need to know where, the reliability of this of the technology. We need to know that it's going to work. There are, I mean, I didn't mention it, but talk about it, but there've been a lot of fads coming along, and we've spent a lot of money looking at all sorts of things which fell over pretty quickly. I mean, I will, you know, I think particularly of bacterial antigens, you know, for meningitis. They were all, you know, they disappeared. And if any of you make a test which relies on IgM, I will personally, you know, it's absolute, you know, so that sort of thing, you know, and so I think these collaborations are good. So when you make, we have a kind of idea which might work, but I don't think that answers your question at all. But anyway, never mind.
You just wonder whether the bar should be lifted for diagnostics and that they should actually have clinical impact studies as part of their approval.
Yes.
Because stewardship, I mean, as you say, it bumps something else out, and we don't even know if it's cost effective or not. Yeah.
I mean, look at COVID testing. You know the bar was really low, and there was a lot of money wasted. You know, on the. You know.
Yeah, and Elizabeth Holmes and Theranos. Adam, do you want to comment on what impact that's had on
no, I think that
disease diagnostics?
Yeah, but she. I mean, it was nefarious from the beginning. So you know, I won't comment.
Okay, excellent. Yep.
Your thoughts on rat tests?
How long you got? So I think, so rat tests have a purpose. There's no doubt about it, and they relieve the lab of a lot of burden to enable us to get on and do more stringent testing for more people at more risk. I think the rat technology will get better, but I think it'll always be flawed. And I think that the fact that you can have an active infection and an absolute negative test, you know, for it's never going to be perfect, but it has a place. Certainly has a place. Okay.
Wonderful. Thank you, Adam. I'll just give you a round of applause. Okay, so next up we've got Simon. Just bear with me. Get the slides going, Simon. Doing a very good job of trying this. Amazing. Okay, so a lot of you will be familiar with Simon. So Simon Corrie completed his undergraduate degree in chemical engineering, PhD in physical chemistry at the University of Queensland. He then underwent postdoctoral studies at the HPV Research Laboratory at the University of Washington, Seattle. Simon joined the Chemical and Biological Engineering Department at Monash University in 2016 to establish the NanoSense Engineering Lab. He is a recipient of the ARC's DECRA Award and the 2018 Churchill Fellowship. And his research interests lie in developing engineered nanoparticles and proteins for applications in biosensing, bioassays, and medical devices. I think this talk will be a perfect segue on from Adam's initial opening.
So maybe, you know, in some ways, following on from Adam's talk, maybe I'll be talking about some of the fads, some of them, but maybe hopefully ways that, you know, we can actually sift through some of the fads and find some of the really interesting stuff. So, you know, just with the introduction from Nanad, I mean, one thing I learned as a postdoc, you know, HPV genotyping was considered at one stage to be a fad, but you know, with multiplex PCR and Luminex type arrays, it's you know, it became a standard, I suppose. So today what I'm actually going to do is I'm not going to talk about my research. I'll put one or two slides in just to give you an idea of what we do. But I really wanted to talk a little bit about the opportunities in CAMR to develop new technology, if we wanted to, for things that make sense to develop, and not to do the Me Too stuff that a whole bunch of companies are doing. Because I think, you know, as a researcher, I don't want to do what the companies are doing. I want to look a bit beyond it. And if there's technology to commercialize, of course we should. And this convergent design is something that I started hearing more about, especially during the pandemic and when the pandemic hit. And I think it's a much better way of doing research. Just a bit of an overview. My lab spend most of our time trying to take these assays, bioassays and biosensors, and put them into a simpler way of being able to be used. And, you know, sometimes we dream up all sorts of new ways to do things. One of my colleagues in New York, you know, came up with this image of, you know, what if we could do sort of real-time biosensing in vivo for a whole bunch of different markers. That technology is actually getting closer and closer. There is a company in Melbourne who's actually doing work on that right now. So we spend a little bit of our time working in blue sky sort of in vivo biosensing, trying to monitor analytes inside the body. And we also design a whole bunch of tools and components and assays for, you know, in the in vitro diagnostics area. And I guess, you know, what we found through the pandemic was it's kind of, you know, the public started to become a bit more aware of diagnostics, I suppose. But also, yeah, I mean, the rat tests as well, you know, they haven't fundamentally changed that much since they were introduced in the 80s. But if they're going to be used a lot more, and I think I agree, I mean, there is a whole bunch of technologies, at least in the academic literature, which, you know, some work and some won't work next week, but they can be more sensitive for one start. But scaling them is the problem. So, you know, convergent design, I'm not a particularly deep thinker. So the things that I learned from convergent design make a lot of sense to me. You know, the idea here is you gather people together who actually have a challenge or a problem and you talk to them. You brainstorm some solutions. The idea is that you identify a gap. You don't, you know, do what maybe I did beforehand, which was, you know, sit in my office and read, you know, great review papers and try to come up with what the problems are to solve. Whereas the gap, there's companies in this space who are developing those, you know, sub 24-hour AST and ID type technologies from whole blood or from cultures. That, you know, there are companies doing that now. There are big companies who have some solutions in that area. Do we need to address that, or is that company with 100 people in it, or, you know, thousands of people in it, going to solve it in you know, much faster? Avoid coming up with the solutions before we understand the problem. And so that means getting all of these people in the room together to talk about these things, and at the very least to narrow down what we can do. Perhaps it's just all adoption. Perhaps the technology is out there and we just adopt it. And the other thing I think, which is also important in avoiding the fads, and certainly in the research that I work in, the field like nanotechnology, biotechnology, there are a lot of fads because things just aren't scalable. If I can make a nanomaterial that amplifies the signal of a rat test in the lab, but it takes a week, 2 postdocs, you know, 10 steps, and I can make a gram of it, maybe, and when we try and scale it, it's a totally different problem. Well, that's just not going to work. It doesn't have to be, you know, redundantly simple. It can be complicated, but it needs to be scalable. And I think in CAMR, we do have the perfect mix of expertise to actually look at these things and try to see where the gaps are. So this was kind of I thought about Nenad's final slide, if you saw it, was a head with all these ideas coming out of it. So that's how I feel half the time. But what I'm saying here, key technical challenges for AMR diagnostics, is more like what I think about. These are the things that keep me awake at night, whether they should or not. But I wanted to put them up there and see what people thought. One thing is, as I said, there's so many problems to solve around AMR. You know, if I think about some other work that we do is getting into is detecting pesticides in agriculture settings. Well, there's a limited number of pesticides and you just develop assays for them. But here, there's all sorts of different problems and they have totally different solutions, and the breadth and depth on each is very different. The sample, you know, is it whole blood? Is it culture? Is it urine? Is it something else? Is it a device surface? Is it a surface in a hospital somewhere? And when I think about sensitivity and specificity, I'm thinking about it from right at the start. So I'm thinking about the analytical specificity and sensitivity, even though I always get asked about the clinical sensitivity and specificity of some of our stuff. Most of our stuff doesn't make it to those trials because it falls apart or something beforehand. But, you know, as one example, when we sort of did some estimates and calculations, and people have, you know, published these numbers as well, for particular bugs, when you can convert CFU per mil to genome copies per mil or something like that as an estimate, you're talking about maybe 1 to 50 cells per mil in whole blood of a pathogen that's in terms of a bloodstream infection. Well, if, you know, using Poisson statistics, counting statistics, that means that in that mil or 5 mils of blood, it's not even absolutely certain that you've got one pathogen in that sample. That's a problem. If we could get more blood, that'd be great. Analytical specificity, as I said, related to the top problem: broad, targeted, diagnosis, AST, both. You know, there's a lot of questions there. Time, as Adam said, you know, I heard from we had the CarbX talk last week, and Richard Alm actually said, you know, I don't think there's any need to develop technologies that could, you know, that actually can be intervening before the first blood draw. You know, maybe that's just always going to be too soon for clinicians to want to act on that, and maybe the empiric therapy will be potentially more important. But before second blood draw, 4 to 6 hours, it gives us an idea of the timing. Or if it's longer, is there a point in developing a new tech, or do we work with the lab to try to improve something if it needs to be done? And of course, you know, it's got to be, I think it's got to be able to be integrated into an existing workflow, whether that's a clinical workflow, whether that's you know, a workflow involving point of care outside of the hospital. And ethically, we should identify things where, you know, identify priorities where linking a result to a decision-making opportunity is paramount. That's got to be there too. So I just thought I'd cover a few. I had a lot of problems narrowing this down because there's lots of tech out there, but I just thought I'd identify a few opportunities for collaboration potentially, because some of these technologies exist at Monash and nearby. One is molecular imaging. Not necessarily always thought about in the context of infection, but another is ultrasensitive assays, and there's just a huge plethora of these around. Many of them I think are fads, but there are some that are interesting there. And I'll just talk briefly about nanomaterials, you know, as a whole, because again, in Monash and nearby, we've actually got quite a lot of experience here. So, you know, there are some, I'm just picking out a couple of neat technologies from the last 10 years or so. This is a technology from Molly Stevens' group at Imperial College. And, you know, this is talking about working on an existing workflow. This is an ELISA assay that has, you know, can detect, this is P24 from HIV, down at 10 to the minus 18 grams per mil. It's crazy. What they've done is simply replaced a reporter enzyme with a catalase and use that to control the peroxide levels in the sample, or in the well, so that you can grow or not grow gold nanoparticles. You get a different color very sensitively. You get another color using the surface plasmon resonance. Again, I haven't seen any other publications after this one. So maybe it just didn't work. You never know. But there's many, many examples like this where you can get down to very, very sensitive assays. And there's a number of people in Australia working on this. Totally off the wall idea. This is actually from Sangeeta Bhatia at MIT, who's a clinician researcher. And they have designed what they call synthetic biomarkers. And what they actually do is they take these nanoparticles, they attach peptides, which are protease substrates. These particles are injected systemically, so they circulate around in the blood, probably for a few days until they get taken out by the liver. But those peptides are cleaved, and the peptides are small enough that they get cleared by the kidneys. And if they're labeled with a certain, either they do a lot of work with mass spec, but they've also shown that if you label the peptides with a fluorophore or a chromophore, you can actually use the urine samples to read out the protease activity. And they've developed technologies for both infectious diseases and cancer. So it's an in vivo detection of in vitro detection of an in vivo process. And these guys are going gangbusters. They've developed a lot of this technology, and have, you know, they've got registered clinical trials, at least on clinicaltrials.gov. Whether that means anything or not, you never know. Molecular imaging. So I just wanted to come up with just a couple of examples. This is an example from Sam Gambier's lab. Unfortunately, he's no longer with us, but this lab developed a number of sort of cutting edge molecular imaging tools for different applications. And this one is an infection application. And basically they've taken a dye and attached a sugar that is not metabolizable by mammalian cells, but is by bacteria. And they showed that they could actually, after injection of this, they could use fluorescence or photoacoustic imaging, which allows somewhat deeper tissue imaging, to actually identify infected muscles. And you can see, you probably can't see on the slide, but you can actually see the foci of infection with the photoacoustics. Photoacoustics is used clinically in 2 places that I can see so far, one in Germany and one in the US. It's co-localized usually with ultrasound imaging. And we do have a brand new instrument to do this work at MBI at Monash. Another one, which is a colleague of mine, and Heather Clark at Arizona State, has done a lot of this in vivo sensing and molecular imaging. And I think the interesting thing about this work is, rather than just imaging a particular marker in a tissue, they can actually measure the level of a particular analyte in that tissue. And that's some of the work that we've tried to do as well. And these guys, in this case, it's not an infection case study in this case, but what they've got is these particles that can detect acetylcholine in, these are in rat brains, not just the level, but also the spatial distribution. And this is the sort of technology that they're pushing through as well. Again, it's an imaging system, and these guys have actually developed a bunch of different detection modules as well, from fluorescence, MR and PET as well, for these types of systems. Again, MR, PET, this is all available at Monash through MBI and also at the Alfred and other places. When, you know, at the moment, the interim head of MBI is Professor Christoph Hagemeyer, who actually works in these sorts of areas as well. Nanomaterials, I mean, for those who are not that familiar with them, nanomaterials have been developed and used in, in biomedical research and developing drugs, primarily in cancer. But the idea is, while there's a lot of hype about targeting drugs to certain areas and all this sort of stuff, I think the clinical reality now is, for the number of drugs that are approved, it does seem that the biggest advantages of attaching a drug to a nanomaterial, for example, is to reduce frequency of doses, to reduce off-site toxicity, because you can change and shift the biodistribution PK/PD. People talk about targeting to tumours and targeting to this and targeting to that. A lot of that has not come to pass, but certainly being able to use relatively toxic drugs in different ways is certainly still available. And the flip side of that is potentially possible for sensing and also, of course, imaging. And there are lots of groups in Australia and around the world developing nanomaterials to help with imaging applications as well. I just wanted to mention as well, in terms of nanomaterials, there's a lot of work, and actually a group of groups in Australia, clinicians and chemists and engineers, who are painstakingly looking at different nanomaterials, or the same nanomaterial presented in different ways, and painstakingly working out how these things interact with, for example, the first thing they're going to see in the systemic injection, which is whole human blood, and looking at the individual cell interactions, whether the cells attach to the particles, whether the particles activate something in the cells. And a lot of this is with Stephen Kent's group at University of Melbourne, with a whole bunch of different chemists. And we've also jumped in there with Professor Anna Travin. We also jumped in there and started to look at how our nanomaterials at least interact with C. albicans. So again, there are lots of opportunities here for collaboration. Very quickly, we've been looking at, since the pandemic, we jumped into repurpose these blood typing platforms. This is just to tell you a little bit about the stuff that we do. So we repurpose these blood typing platforms, little gel card tests that are available and supported in a lot of areas around the world, to turn them into serology tests for COVID antibodies. It wasn't really required that much in Australia, but in places where PCR labs were not available, some of the people have at least taken the papers that we published and sort of set up some assays like this. We also think that these agglutination assays can be made much more sensitive with the nanomaterials that are available in the ultra-sensitive assays, etc. And I'd love to do more about that. And my colleague, Professor Gilles Garnier, has also worked with local companies to produce these blood typing cards that you can just use at point of care. We also work in engineered proteins and antibodies. We don't just affinity maturate and change the activity and things like that. We also do funky things like whack in dyes into these reagents so that when they bind a target antigen, you get wavelength shifts or intensity changes of a fluorescent dye. So we're trying to make reagents that can just bind to a protein and give you a signal in one step. And finally, we're also working in these nanoparticle-based imaging and sensing area. And so, for example, one example is we design these particles that are active in ultrasound, and they can actually report the local pH in a live animal model. That's and we're kind of pushing this forward as well in photoacoustics and other things. Fluorescence we do a lot of work in, but fluorescence is not going to be all that translational. So just in summary, in terms of opportunities that we're working in, re-engineering established assays. I mean, that seemed to be quite useful in the pandemic. We wanted to do something, and we found some clinicians who thought that was going to be useful. Molecular imaging and sensing, there's lots of groups who are working around this at Monash, but very few applications in infection or let alone AMR. Phage, we've got so much work on phage. There are reporter phages out there for sensing, imaging, diagnostics. I wonder if that is actually a pretty untapped area. I found some papers. The best paper I found was still in bioarchive, so I don't know what that means. And leveraging, I think there's a lot we could do to leverage the in vitro screening and in vivo models work that Chris and Tom are doing, and I'll talk to them. I can see Tom down the back there. I can't see Chris, but maybe he's online. But we could certainly take more advantage of that now that we've got those up and running. And convergent design, I really have enjoyed this approach of going and talking to people. So often you find actually there's not a challenge that we can jump into. That's fine. It just means that we move to the next one. There's always challenges that we can look at. But that's all I had to talk about today. I'd just like to acknowledge the people who have done the work at Monash and in other places, and the funding, and which includes CAMR, which actually funded our work during the COVID pandemic, which we just wouldn't have been able to do without funding from CAMR. Thanks very much.
Excellent. Thanks so much, Simon. That was a wonderful overview. I think we have time maybe for one question, and then we might move on. Anyone? Yep.
Thank you very much. I've really enjoyed this. I'm just wondering, there's so many technologies that people all around the world develop, and very few of them actually make it to the market and make it to the patient and, have an impact. Can you tell us a little bit about what is your opinion, why that is the case, and can we improve this in the future?
I don't know about you, Helmut, but I'm an academic. I often start from looking at what people have done in the academic literature. I'm not convinced that that's the greatest place to rely on, because, a test that someone develops and publishes, you have no idea how reliable it is. It may be, it's not useless. It's shown some useful trait, but you just have no idea how reliable, robust any of it is. I also think that as academics, sometimes and my supervisor was like this too as a student, you'd develop a test to a certain point, and then they'd kind of get bored with it because they'd found something else. And so it never got into any type of clinical trial, or even just testing on real samples. So, you know, trying to identify what technologies are robust and what technologies are fads, or, you know, as Adam said, or just not going to stand up to the rigors is pretty important. I think there are examples of quite complex techniques that really are robust, but I think you got to find them. Yeah.
Thanks so much, Simon.
Because we can't hear you.
Oh, really? Down the back? Yeah, yeah. Sure.
If you could just speak in.
Yep. Like this? I think we need to get pretty close for it to be. Yeah.
Yeah. So
is it possible to turn it up?
Yeah.
Yeah.
It's really getting to the back of the room.
Okay. No worries.
All right. Thanks.
Okay. Wonderful. Well, thank you, Simon. Now, welcome Dr. Hemut Thissen. Dr. Thissen leads a team at CSIRO Manufacturing in Melbourne that is focused on new and improved biomedical devices, and in particular, the effective control of biointerfacial interactions. He joined the Premier Australian Government Research Organisation after completing his PhD at RWTH Aachen University in Germany, where he also started to translate biomedical research into the clinic. While he has published more than 180 journal articles and book chapters, his strong translational focus is reflected by his role as the director of CSIRO's Biomedical Materials Translational Facility and his contributions to multiple successful biomedical products. Importantly, he has served his scientific community in many different roles, including president of the Australasian Society for Biomaterials and Tissue Engineering, and has been a mentor of many early career researchers. Thanks so much, Hemut.
Thank you very much. So can you all hear me at the back? Yeah, excellent. Okay, thank you so much. And thank you so much for inviting me here. I really appreciate this. This is a great opportunity, and I could not agree more that how important collaboration is from our point of view as well. And I really, you know, I really, you know, support this. Of course, I hope you all know what the CSIRO is. That's, of course, the Australian National Research Agency. Our role, of course, is very different from university. And of course, we are mainly focused on translation, on helping and assisting industry. And therefore, we are very much focused on intellectual property, on working with industry partners. And so, you know, our research focus is different. And our, you know, the way we see the world is probably a bit different as well. One example is that we are by far Australia's largest patent holder, and you know, that emphasizes our you know, our view of the world and our you know, path to impact that that we think is important. Of course, we are also you know, given how focused we are on government priorities, very much interested in antimicrobial resistance. Of course, we have many collaborations with several people here in the room, including the clinicians, and you know, CSIRO itself has also recently established a grouping within the organization, the Minimizing Antimicrobial Resistance Mission. And you know, we hope that many good things will come from this, including enhanced collaboration. I'm going to, you know, as the previous speakers, will tell you a little bit about my own view of the world, of, you know, how, you know, biomedical devices can really contribute to this area, this issue of AMR. And, you know, I think, you know, the main message that I want to convey here today is that really interfacial interactions, so interactions between materials and the biological environment, are really the key, from my point of view, that determines, in many cases, not only on biomedical devices, the you know, clinical performance, the clinical outcome, but that is also a very, very important issue for AMR. And you see here some examples for biomedical devices ranging from biosensors to implantable medical devices. You will see that they all have they all consist of different materials, polymers, ceramics, metals, etc. But they all have a surface, and again, that surface will provide an interface to the biological environment, and that is really very important to get right for improving the clinical performance and the function of these devices. Now, I'm going to talk about both biosensing a little bit and also about medical devices. First of all, I would like to say that in the biosensing field, we have recently looked at what should we really do into the future in this field. And of course, you all know that AMR is not just an issue for the clinic. It's an issue that occurs equally in the environment, in agriculture, and basically everywhere. So we all know that if you have AMR somewhere in the environment, well, it'll soon be in the clinic as well, and vice versa. So there's all sorts of routes of transmission of AMR genes, as you all know. But it means that we have to not only survey and surveil AMR in the clinic, we also have to do this in the environment and in agriculture. And if you look at methods that you can use for biosensing, well, then you can very quickly you will see that the challenge is often to achieve sufficient sensitivity as well as specificity, and that the challenge doesn't come just down to developing a biosensing method, but it's actually working with complex biological media, and that can be sewage water, it can be blood samples. They're all at the end of the day complex biological media. And you have to work with this. So there's multiple challenges there. Basically, if you look at these environmental samples, the agriculture samples, and others, our thinking was that really you will likely focus on DNA. And of course, as the previous speaker was explaining, you have these very basic steps of signal transduction and signal processing leading to an outcome and leading to a reading. We have these different domains, and of course, these different domains are associated with different challenges. For us in CSRO, that means that people in the agriculture space actually develop their own sensors, and we in the biomedical space develop our own sensors as well. So that's probably the wrong approach, and through collaboration again we are trying to improve on what we're doing. But the simple fact is that we have different priorities. For example, in the environment, cheap autonomous sensors are required, whereas in the healthcare space,
and he was like, I could give you these glasses, and he took one, and I was like, oh, that's nice, but it's not enough.
we have I'm not sure what that was, but we of course have in the healthcare biosensing space, we have different requirements. Again, importantly, these interfacial interactions, including very importantly the issue of biofouling, play a very essential role here, and that's where we can really make a difference. The biosensing techniques that we in our lab use are particularly focused on, for example, nanostructured materials, and we work very closely with Professor Nico Völker on developing nanochannel based sensing technologies, again focused on in particular the DNA sensing. They have many different advantages. I won't go into too much detail what these advantages are, but it'll suffice to say that you know, we are in a lucky position, and the previous speakers pointed this out as well, that we have access to excellent infrastructure, and in particular here in the nano technology space, the Melbourne Centre for Nanofabrication, and that is really key for, you know, moving forward, that we have this. Also, importantly, you know, for industry, from an industry perspective, it is very important that these facilities operate under a quality management system, and that is the case here for the MCN operating under ISO 9001. When we think about medical devices, you know, we reviewed a few years ago, you know, what are the infection rates associated with different medical devices over the lifetime of these devices, and you know, you will see that some of them, like contact lenses, you know, you probably can't improve this much further. So they're very low incidences of infection, whereas others really stand out. And, you know, something that we have collaborated with our colleagues here, ventricular assist devices, they have up to 80% infection rates on these devices. That's really totally unacceptable. But urinary catheters are also a fantastic example. You know, they create a huge problem in the healthcare space. And if we could just address this one, you know, this one device and make it better, that would have enormous impact. And that would not only have enormous impact on, you know, on improving, bringing down healthcare costs and improving, you know, patient outcomes. But of course, this is all related to AMR as well. And so, you know, when you think about these, you know, catheter-associated urinary tract infections, that's actually something that we are, you know, focusing on in our lab. You know, this is on top of the list really for hospital-acquired infections and you know, how people actually get these. Just to give you an example. So, what sort of antimicrobial technology options do we have when we, you know, look at these interfaces? How can we improve the outcome for our devices? Well, you know, we have a few different options, you know, that we prevent attachment due to polymers at that surface that provide low fouling surfaces. We can have contact killing activity of the surface. We can have, of course, have release mechanisms. And we can also have topography that leads to contact killing. In particular, what we're focusing on is the combination of different mechanisms to provide an improved antimicrobial protection. And when we think about, you know, some of the recent work that we have done that we are really excited about, it's, you know, processes that are really one step. So, you know, when we talk to our industry partners, you know, the process that we use for modifying a medical device, it's got to be one step to be able to be integrated in a manufacturing process. And often when you think about surface modifications, these are, you know, multi-step processes where you have to activate the surface and deposit something, etc. So with these polymer systems here that we have developed, these are stable polymer coatings that are, you know, UV, and we, you know, we can, you know, use these polymers that we have created to provide, for example, low fouling properties and a, you know, radically polymerizable, radically UV reactive group here on the right, and this allows covalent cross-linking of the polymer on a surface in one step, and at the same time, this allows covalent cross-linking with the substrate. These ultra-low fouling, you know, coatings that we can provide on surfaces, you know, if we get the right molar ratio of these components, can completely prevent cell attachment and also, you know, bacterial attachment. But of course, we can also integrate into this antimicrobial compounds, and, you know, examples for this that we're exploring includes, you know, antibiotics, quorum sensing inhibitors, etc. also Fan Yi is in the room here working on again multifunctional antimicrobial coatings by combining these ultra low fouling polymers with selenium nanoparticles, and again these can be deposited in one step, and they are quite effective in combination. This multifunctional coating can really provide highly effective protection from attachment and biofilm formation on these devices. If you do a scratch test, of course, on a low fouling coating, then you will find that bacteria will attach on this scratch. And we have to be honest, if we think about medical devices, they will be handled, and there will be issues with non-uniform coatings and scratches, etc. So we have to be able to deal with this. And so if these particles are actually incorporated well, then that provides protection not only of the actual surface where the polymer is present, but also in its vicinity. And that's a very important point, that can be achieved by this kind of combination coating. It's also important to point out that if we talk about these new combination coatings using selenium as an example, this is has a therapeutic window. Actually, that's very, very important in comparison to silver nanoparticles, for example, that have become very popular, which of course have a toxicity to mammalian cells that is basically at the same concentration. So these selenium particles perform much better and provide this therapeutic window, if you like, to achieve what we want. Last not least, I will also give one quick example to say that we have also the opportunity to provide nanostructured surfaces, and you may all know that insect wings have demonstrated that basically there are some nanostructures on there that provide mechanobactericidal activity. And this, of course, can be repeated in medical devices, on the surface of medical devices. We have worked, for example, on titanium as a very popular material, and with a very simple process, you can, perhaps not create, very controlled structures on these surfaces, but you can definitely, create structures on that surface that will provide this mechanobactericidal activity. Again, a very simple one-step coating, and of course this will kill bacteria in direct contact, and really this technology does not care if your pathogen is a superbug or not. It'll have the same mechanism and it will always work, and we have demonstrated this. So with this, I again want to point out that we have our own facility, the Biomedical Materials Translational Facility, to closely work with our industry partners on bridging this translational gap. And again, we are working under a quality management system here to be able to, to do this, to prepare for, taking technologies to the next level, to go to a regulatory agency and say, we have done these experiments under these conditions, so that you will accept these data. That's very, very important. So again, the infrastructure is extremely important for us and for moving forward. So in conclusion, again, I hope you agree with me that biomedical devices, ranging from biosensors to implants, are expected to play a significant role in the fight against AMR. I have to say they're often overlooked, really, and that the effective control over biointerfacial interactions is really a key to future developments. Thank you so much.
Wonderful. Thank you, Helmut. I've been a little bit remiss in my role as the chairperson as we've gone over time. So perhaps one question for Helmut directly, if anyone has one. No? Okay. What we might do is actually move on to the panel then. Can I just invite the speakers to come forward? Excellent. Thank you all for putting together such a wonderful panel of talks with very specific perspectives, and I think it really kind of captures a whole range of issues around AMR diagnostics. I guess one of the things that kind of kept coming up is this sort of challenge of translating things to the clinic, and I just wanted to draw you out a little bit more on this and just ask about sort of examples of things that have been happening recently where we have actually seen novel technologies reach clinical practice. And I think I might sort of just start with you, Adam. I mean, a lot of us work in the genomic space, and I've found it quite interesting that labs are now starting to actually have some of that genomics work accredited. Do you want to comment a little bit on what that process is like? What are some of the, achievements? What have been some of the challenges, and how you see the path going forward?
Yeah, thank you. It's a massive topic, and it's of huge interest to us. I think I have to say, as far as we are pathologists in the lab, and we are under the auspices and we are accredited by a college. And there is some degree of trying to catch up with the technology into how we can actually put that into the clinical space in our labs. And I feel, there's a whole older people like me who, in our career, this has come up, wasn't a problem, wasn't a thing to begin with, but now it's very much there, and we have to learn. And so I think we've relied for a very long time on the reference labs to do that work, and so sending them isolates and to do all the analysis, the sequencing, the bioinformatics, but we now have to do that now. It means we're going to have to change our structures. We're going to have to get a whole load of different specialties within the lab. I mean, we really are scientists, technicians, and a few doctors, but we're going to have to change that. So, I'm not a lab I don't have a lab who actually does that yet, but we're certainly looking to do that. But and I think we need to have collaborations, particularly with departments like infectious diseases and with bioinformatics and with epidemiologists, and we all need to basically sit in the same place to be able to do this. So I think we have been slow, but we need to accelerate and get it done. And I think there are movement to do that. So
did you have a comment, Simon? Or?
Oh, no.
Not particularly. Well, yeah, I think the regulatory aspects are something that kind of came across in all of the talks, and I was just wondering whether you could comment a little bit on how that impacts some of the work that you're doing, and how you think about what things should be moving forward and can move forward.
Well, I'll start, but I just had a perspective from watching the explosion of COVID rat tests that became available in the first 6 months or so of the pandemic, and I mean there was at some stage there was like several hundred companies who were trying to put forward tests. Many of them had never produced these tests before, and of course at Melbourne Uni they were trying to evaluate these for TGA and trying to catch up, and then that doesn't even include the kind of clinical impact studies that Anton was talking about. So, I mean, for us, we were thinking about it with the COVID work that we were doing, but but our approach was really to link in the clinicians and the lab staff who were potentially going to be running these tests and try to get them to, stop us at the start and sort of get us to do things in a certain way. But then I'm sure Helmut has another perspective as well.
Yeah, I think, again, we're talking about collaboration here. I think it is a certain mindset that you will need if you are working in quality managed environment. And I think it's very important that we keep this in mind for education of the next generation of scientists. I think that's the key, because, it is really important. We have had our own examples where we have developed technologies, and got very excited about this and wanted to go into a clinical trial, and were basically told, well, where are the data that you have produced under a quality management system? Well, we haven't done that. Well, then you go back to square one and do all the work again. And after, having this experience, it becomes clear that what you need to do. And of course, I think the education aspect for me is key, and of course having the facilities available to actually do this. And if we've actually done this. We have interviewed many companies here in Melbourne, in particular, working in this space, and it's very hard for these companies to find the right facilities and. You need certain, you need a clean room, you need, to work under a certain quality management system, etc. It is hard to get to this. So, I think we know what the issues are, and I think together we should address this.
Can I just add something? So when we have talk about lots and lots of rat tests coming all at once, there is a big power of those bigger companies who pre-existing to push their product forward, and maybe very good technologies might be pushed aside because it's overwhelming, and there aren't the regulatory authorities or the staff to deal with it, and so you find the big names will push their product forward because they have the actual structures to do that, and maybe we miss out on good, particularly local. Because we'll always look to the international where they've had those, even a little bit of clinical, trial data will push it over the edge. Some versus none will out.
And that's a really good point. Any questions or comments on this from the audience? And Tom?
I was just wanting to hear your thoughts about that kind of preclinical pathway, and whether you think there are hurdles, particularly, I guess, in that sort of animal model space and the device infections. I know that we're trying our best, but it's a really challenging sort of area around what will be acceptable for that translation pathway at an animal model level for some of these device infections. Yeah.
For the way, yeah. For us, this locally, this is a challenge. Having access to preclinical models, and we've tried with a couple of PhD students to get those happening locally at Monash, and we couldn't. It was just challenges of everyone doing all sorts of research in the same place, and using the same sort of equipment and not wanting to contaminate it between things. That was a real challenge. So most of our in vivo work has been on totally different disease systems that are not infections. We would love to do more. We would absolutely love to do more. And there's a need, and there's technologies that could be adopted from other areas and used there. So if we could solve that challenge, and maybe it's a local challenge, I think other places do it a bit better, but if we could solve that, then it would be pretty great. Yeah.
Yeah, I think I agree. And I have to say, it is hard. The translation bit is very hard. So you really have a fantastic, early stage, research community here in the country, and great research outcomes and facilities and everything. But then the translational bits, that's, we have to work on improving that. Yeah.
Yep, no worries. Kwong?
Thanks. Just a comment and a question. So it's around when your technology has been commercialized, and one of the barriers, from my opinion, is that if it's the uptake from the GPs or the clinicians, if it's not on the Medicare beneficial scheme, so it costs a lot for the patients or for the clinician to order for the patients, and sometimes they don't put that burden cost onto the patients, so they're not ordering the tests or that kind of test. Can you comment on that? How can we solve that? Is there a better way of doing things?
Oh, look, massive problem. So, I mean, there are limitations to the number that are MBS rebatable, the number of tests, and there is a whole. Part of the problem is that they can't let everything through, otherwise it'll blow an already massive budget, because you will often these tests are more expensive, and that they aren't. They're more niche, and so they just don't fit they don't fit the budget. I think the other thing, it leads to also, I worry about a degree of unscrupulousness as well. Is that they've got to be able to. I mean, it's getting people to use these tests that are not MBS approved. I think it will. So sorry, what am I trying to say? So while that's a great process to get them approved and get them on, they usually work very well, and they give benefit to society. What has happened is that we have pushed a whole load of people who may have had good ideas one way. We've pushed them away, and there's a whole bunch of unregulated tests going on, which are costing people thousands of dollars each year. Which, and I think part of the problem is reining them in as well. But basically, it comes down to money and allowing us to have a wider suite of well-regulated testing. So I don't know if anyone else.
Thank you. Katrina?
Thank you. So this is a separate question, although, yeah, the whole PBS recognition of point of care tests is a really huge issue as well when it comes to things like STI testing in remote communities when there aren't alternatives. So this was a question for Helmut. I just noticed in one of your slides with your anti-fouling surfaces that there was inclusion of a really broad spectrum antibiotic that has a marked propensity to develop AMR, so ciprofloxacin. So what is the thinking and discussion in the field when it comes to surfaces that prevent biofilm attachment or development and infection, about inclusion of antibiotics in these surfaces? Because it seems a very time-limited strategy that in fact could backfire.
That's a great question. I probably I must admit that I think the awareness of AMR is very good by now in this field, in the biomedical device field. And I, of course, absolutely agree with your comment. It's not a good strategy, but it's something that serves, an in vitro assay, because it will tell you if it works. In terms of, what sort of antimicrobial compounds you can use in the context of devices, yes, I absolutely agree that antibiotics are not the first choice, because of the reasons that you mentioned. So, again, it's an example for, demonstrating a concept, but, it's not a good choice if it comes to the final translation. Does that answer your question?
Excellent. Thanks so much. Any other questions from the audience? No? Okay. I might kind of keep going on this sort of track, and I think, Adam, you put this in a very nice way about AMR diagnostics not just being about the lab and moving out of the lab. And I think, we're starting to talk a little bit about point of care testing. And I, was hoping you could comment a little bit on what you see as a panel from that perspective for AMR diagnostics. And also, I think the other kind of question that you raised, Adam, was around equity and access to some of these sorts of tests in, I guess, low and middle income sort of settings. And are these sort of factors that come into play in terms of the work that you're doing?
Thank you. Thank you. So the point of care testing is really important. I think the point of care testing gets to a whole bunch of groups of people who have particular resistance patterns and that we don't really see. Our data seems to be mainly represented from labs that are, big hospital labs and sometimes private labs, which are more of a community-based infection group. And there are whole populations missed out. But the difficulty is that, as you know, with a point of care test, how sure you are that is the right answer. I mean, and you are, and also with the self-testing on top of that, how do you collect that? How do you get, how do you know it's all, so as long as it's simple enough, as long as it's robust enough, and that there must be some way of collecting what result you get it. That is very, I think we can really get much more information about what the problem is, where the problem is, and how to address it. In terms of equity, I mean, I think the cost thing is really important, and the availability, and we have actually taking not only just a point of care where away from a laboratory, but taking it out beyond that to people who can't even access that. I mean, people are not going to GPs anymore because none of them are bulk billing. So in fact, taking it out onto the street and actually going and finding people and then getting information about them, because they are the most vulnerable. And then take that globally, and you've got the poorest nations in the world who are not in any way immune from this. In fact, many times they are the most affected by AMR.
My only perspective was when the rat tests were the front line through COVID. I mean, to me, looking at the rat tests as someone who comes up with new ideas all the time and tries them out and things. The rat tests fundamentally haven't really changed since the 80s. And I wonder if there's opportunities, through talking with communities like this, to come up with a completely different way of doing it. There are really old technologies that got surpassed by monoclonal antibodies and ELISAs and things like that can actually be repurposed and reimagined. The funding agencies don't like it. It's all, this is old stuff. Why would we look at that? But actually, I think there are some other ways of doing things that don't rely on those methods.
And can I add to this that the medical device community has certainly realized that how much waste is being produced, and that's a prime example, for these tests, and you just throw it all away. So there's many opportunities that are also associated with this.
Yeah, we have a fourth year student at the moment doing a full life cycle analysis on the rat tests, and it's just mind-boggling, the amount of polystyrene and polyethylene that's just chucked into landfill. And of course, before it was used by everybody for several years, constantly. It may not have been such a big load, but it is huge. Yeah.
Thanks, Simon. So back to the future, huh? Is that the paradigm? Okay, I think I'll just ask one final question, unless someone has a question in the audience. Okay. So one of the things that I found fascinating, I think, again, several of you touched on this, how we can kind of also take lessons from other fields. In particular, sort of the cancer space, I think, has a lot of informative lessons, both in terms of diagnostics and therapeutics for AMR. Can you comment a little bit on, what we can draw from that and integrate into the sort of work that we're doing?
Just from a very broad perspective, I mean, a lot of the work around nanomaterials, nanoparticles for delivery, sensing, imaging, etc. A lot of that work, the very few products that are available are almost all in the cancer field. Of course, there's also some antifungals that are liposomal antifungals, but there's been a lot of hype, and there's a big hype cycle around that. But I think now we've got a much better idea on how those things work, and so it's a good time to look at expanding that to other fields, because we know a little bit more about how technically how they work and how they don't, what's real and what isn't. So it's rife for change, I think. Yeah.
Yeah, I think it's not just the cancer space. I think it's many different areas. It's as I said before, it's the the environmental space, the agriculture space. It's so we've got to communicate, and we've got to collaborate to learn from each other, and it's that opens opportunity for taking technologies, but also better integrating what we're already doing in these different fields.
Yeah, I think closer to our home, I think I mentioned it about pathology stewardship. I think it's important that we focus on what is important, because of the way we are funded and the way government pays for pathology and all that. I think we really need to say, what tests are we doing that are just unnecessary and repetitive and useless, and focus on what we need. And so things like AMR really miss out if we don't have that stewardship. And so, antimicrobial stewardship, I know it's a sort of, our friends right there, but what they, made great gains in that, and making it the norm rather than the exception, I think.
Thank you. Excellent. Thank you. Any other questions, comments from the audience? Yeah.
It's very, very quick. The concept of repurposing drugs is very well accepted, and largely because the pipeline is so costly and lengthy. But really what you're talking about is repurposing existing diagnostic technologies. And if you combine that with a cost effectiveness analyses, then you can really make a pitch to funders that repurposing existing diagnostic technologies is cost effective and the way to go. And it's obviously really important to not keep trying to reinvent the wheel, because a lot of things do get passed up.
Yeah, thanks, Katrina. Okay, well, we might draw the session to a close. If you could join me in thanking the speakers today. I think it's been a wonderful start to the day. Thanks. So what we might do, we are running a little bit behind schedule, but I think we'll try to reconvene at 11:15 for the next session.